Today, we're looking forward to an interesting and positive debate. We are aware that many of the issues we are discussing today are contentious and they tend to polarize discussions. The aim of today's discussion is, however, to be informative and to show how different regions of the world have thought about this process. With the European Commission having started the process to think about how these new technologies should be regulated at a European level, we think that it is very timely to start a broader conversation about what we can learn from other countries' experiences and the broader implication of the decisions that Europe will make in the coming years. We have seen in previous work that the values we think are important to safeguard in Europe, such as supporting small and medium-sized farmers, safeguarding biodiversity, strengthening our rural communities, addressing the challenges of climate change and unpredictability of weather. In understanding regulation within this context as a way to shape how these technologies will benefit society and promote certain values, we hope today's conversation will be informative and inspiring. We look forward to contributions from Japan, Australia, Argentina, India, the African Union, and the UK. We're also glad to have with us today representatives from the European Commission, members of the European Parliament, and the USDA. As we saw in the initial work developed by the task force, whatever path Europe chooses to take, it will have significant impact both in Europe and beyond on how agricultural systems develop in the broader context of sustainable food systems, climate change policy, and safeguarding biodiversity. I would now like to pass the floor to Dr. Peter Kearns, who will be chairing today's meeting. I look forward to an exciting debate. Peter, the floor is yours.
Many thanks, Erica, and ladies and gentlemen, friends and colleagues. Today, as Erica mentioned, we are operating across a range of time zones. So good morning, good afternoon, good evening, wherever you happen to be. We have a pretty tight schedule, so I'm going to try and be brief with my opening remarks, but just to let you know, I am Peter Kearns, Special Advisor with Reimagine Europa. I'm a geneticist by training, and I've had quite some years of experience in official positions in the regulatory oversight of products of modern biotechnology. I am most grateful to all of those who will make presentations today, and when we've had the keynote, I'll come back with an introduction to those shorter interventions. So I think it best that we go straight away to our keynote presentation. This will be about 15 minutes, I think, with about 20 minutes of Q&As, and Erica has already explained how we will take those questions. Our keynote presentation today is, of course, from Professor Jennifer Doudna, who is the president of the Innovative Genomics Institute, Berkeley, California, and of course, one of the 2020 Nobel Laureates in chemistry. So I think that's sufficient from me for the moment. So Professor Doudna, many thanks for joining us today. And if you're ready, the floor is yours. Thanks so much.
Professor Kearns, thank you very much for that kind introduction, and Erica as well. I really am honored to be here and have an opportunity to speak with you. If you could share the slides, please, I will go through a short presentation, and then I would like to save plenty of time for your questions.
Okay, if you can see the slides, you can take the control. You can see the button, the upside. Okay, please. Okay,
very good. So as Professor Kearns said, good morning, good afternoon, good evening. I'm sitting in Berkeley, California right now for this presentation, so it's morning our time. I'm delighted to speak with you about CRISPR and the future of sustainable agriculture. This is a topic near and dear to my heart. I am not an agricultural specialist or a plant biologist, but I think, as you will see, the technology of CRISPR is opening the door to extraordinary opportunities in agriculture, something that our institute is keenly interested in and I think affects all of us as we deal with the challenges of climate change and increasing global populations. On the next slide, let's see, the arrows are working. There we go. I wanted to first introduce you to CRISPR. This is a technology for genome engineering, and it started for me when I began an international collaboration back in 2011 with Professor Emmanuelle Charpentier, then in Umeå, Sweden, and her student Chris Chylinski in Vienna, Austria. So it's been a truly extraordinary and international partnership that led to the development of the technology that I'll describe. This is a tool that allows scientists to make precise changes to the DNA of any cell. What you see in this picture is a representation of the molecules that achieve genome editing. In green, the protein called CRISPR-Cas9. In yellow, it's RNA guide, which tells the protein where to go in the DNA. And once this protein assembles on DNA, hopefully you can see the DNA helix opening and the strands being cut. This is the fundamental chemistry of the system, but to explain how it works for genome editing, I want to show you the video on the next slide. So this is a representation of how CRISPR-Cas9 works in animal and plant and human cells. The protein Cas9 is able to search through all of the DNA in the cell to find a short sequence that matches the sequence of the RNA guide. Once it arrives at that position, the DNA unwinds, the protein is able to introduce a double-stranded break to the DNA, and that breakage triggers repair enzymes in the cell to fix the break, and in the process, introduce a change to the DNA. There are many different ways to implement CRISPR that allow DNA to be removed, DNA to be inserted, or the actual letters of the DNA to be altered. And this provides a profound opportunity now for scientists to control the genetics of animals and plants and to make changes that help us both understand the basic functions of genes, but also increasingly to interfere with the DNA itself for medical or agricultural applications. So when we think about where this technology is headed, it was just under 10 years ago that Professor Charpentier and I published our research for the first time in 2012. Less than 10 years later, we have multiple companies that have started around the world that are working with CRISPR. We have multiple clinical trials that have already announced their exciting results. And importantly for this conversation, many agricultural companies, large and small across the world, are adopting CRISPR because they recognize that it's a powerful tool for today and for the future. So I wanted to briefly mention what we consider the 4 revolutions of plant breeding, starting with selective breeding that required a lot of time and depended on random changes to the DNA in plants to give rise to desired traits. This led to mutation breeding since the 1920s, in which random changes could be introduced using radiation or other kinds of mutagens like chemicals to introduce random alterations to plant DNA, followed by selection for plants with desired traits. Again, relatively fast compared to selective breeding, but a random process. And then since the 1980s, it's been possible to do what's called transgenic breeding. So only the desired gene or trait is incorporated, but that incorporation is random into the plant genome, and that can trigger undesired properties in the plant. And now with genome editing, we have a unique opportunity to make precise changes to the DNA of plants. This process is fast, and importantly, it leaves no trace. So it allows scientists to make the kinds of changes that will introduce desired traits without introducing undesirable features or alternatively losing traits that are of interest. And I personally, although I'm not a plant biologist, I think that the potential impact of CRISPR on agriculture across our planet is huge. I think it has the potential to impact many more people in a shorter timeline than what we will see in medicine, for example, at least in the near term. So when we think about the opportunities with CRISPR, I want to point out that, and I think everyone here is aware of this, that climate change has enormous impacts on our food system. And what I'm showing you here are just, some of the ways in which the changing climate across the planet is affecting the way that we grow food, the way that we distribute food. We have to think about effects on globalization, and I sort of summarized on the right-hand side the fact that there's increased disease severity, lower yields of crops, often due to challenges of the changing temperature and the effects of that, and also lower nutritional quality of food. All of these things are immediate impacts of climate change that we're grappling with, I think, across the planet at present. And these impacts are not in the distant future, but again, I think we're all aware that we're seeing these impacts right now. So there are changes that are affecting, for example, the growth of grapes, the harvesting of olives, the production of chocolate, personal favorite of mine, and of course many other crops that are being affected in different climates and niches across the planet. So when we do think about how we protect small farmers, how we protect family farms and crops that may be important regionally, I would argue that CRISPR will have an incredibly important role to play in that process if it's managed well, both from a scientific perspective, but also from a regulatory perspective. And importantly, the technology is moving very quickly. I can't emphasize this enough to you. This is an extraordinary time in science. We've seen already thousands of publications, it may be tens of thousands at this point, using the CRISPR technology in many different systems. And a lot of that emphasis is on systems that are important in agriculture, whether we're talking about plants or the microbes that support agriculture in different parts of the world. We just saw a recent announcement of the first CRISPR edited food to go on sale. This is the CRISPR tomato. It has supposedly increased nutritional value. This is on sale now in Japan. We saw news earlier sometime in the last year about CRISPR edited beer made from hops that are edited using CRISPR. I've had that beer myself, in fact. And coming soon are crops and other agricultural products that will be the result of CRISPR genome editing, and some of them are listed on the lower part of the left-hand part of the slide. And so when we think about applications of CRISPR that will impact climate change, we have to be thoughtful about how the Earth will be able to accommodate the growing population that we are facing, and also at the same time thinking about how our food system is responsible for more than 1/3 of greenhouse gas emissions. How do we grapple with this seemingly this sort of juxtaposition of an increased population and increased greenhouse gas emissions from food production? And I think CRISPR will have a critical role to play in addressing that challenge. And finally, I want to point out that it's been clear from the very beginning with CRISPR that it has potentially huge ethical implications, not only in agriculture, of course, but also in the treatment of human beings, and in particular, the potential to use CRISPR in the human germline to make heritable changes in humans. And this is not theoretical, it's in fact already happened and has spurred an international partnership to ensure that as much as possible, the international scientific community is aligned in the way that we pay attention to and encourage transparency of the technology as it continues to advance. And I'll be happy to say more about that in the questions if anyone wants to talk about that. And then I just want to, importantly, I want to turn to the public perception. I think that, again, we're all aware that equally important to the actual science and the technology is the way that it's perceived by the public. So I think we all have a responsibility to ensure accurate information that is relayed to the public. Even when there is accurate information, of course, there can be misperceptions, whether they are incidental or purposeful, that can guide the public's thinking about new technologies in different ways. So I encourage all of us to take a proactive approach in the case of CRISPR. And so in conclusion, I just want to point out that CRISPR adds speed and accuracy to plant breeding without the addition of foreign genes. So it's a tool that allows precise changes to be made to DNA in plants. It's widely accessible. So it's not only a technology available to large corporations, but I would argue can be made available to small scale farmers as well. And that will also be an important part of the way it will have global impact, in my opinion. Public acceptance is not assured, and it will require careful stewardship as the technology moves forward. And of course, underlying all of this is appropriate regulation that can ensure responsible use of a powerful transformative technology that I think can benefit humankind if it's used safely. I'll stop there and I'll be happy to address your questions. Thank you very much.
Thank you so much, Professor Doudna. I think you all know how to ask for the floor, and I will be kept updated with questions to come. But I can see immediately that we have Paulo de Castro, who has his hand up, and Paulo de Castro is currently a member of the European Parliament, was former Minister of Agriculture in Italy. So, Mr. Paulo.
Please, Mr. Paulo, now you can turn on your microphone.
Okay, you're supposed to see me now.
Yes.
First of all, you listen me? Great. Okay. It works?
Yes.
Okay. So first of all, thank you. Thank you very much. Thank you very much, Professor Jennifer Doudna. I'm impressed because I'm spending large part of my time in the European Parliament to try to convince that new breeding techniques, these technologies, the CRISPR technology is nothing to do with GMO. You know, we have a lot of pushing people that said that these the same things and don't help us to try to explain that this kind of technology could be the future of agriculture in the sense that they can very much introduce innovation and try to reduce the chemistry and use the genetic instead of chemistry. And that would be a tremendous impact in agriculture. And as you know, the Green Deal and the strategy of the European Commission is trying to push and to involve, you know, the debate in the European Union. It's very much inside of this. But we need you, Professor, we need you to try to explain very clear the difference between the two technology, the old GMO, which introduce gene from one species to another, and these technologies, the CRISPR genetic and genome editing, we doesn't know. And this is crucial for us to try to have a large support of the public opinion, because, as you know, we have a huge number of member states who are against GMO, and of course, because the public opinion is against. So please try to give us the clear idea how different are these two technologies to explain to the people. And again, thank you very much for your incredible important job.
Professor Doudna.
Thank you for that question. I would say that what CRISPR does is it does what happens in nature, but a lot faster. So it essentially allows scientists to accelerate what would normally happen over many decades of plant breeding, for example, but it allows that to happen quickly. And furthermore, it's a tool that has precision. So rather than relying on random changes to DNA, it allows precise and accurate changes to be made in one place or a few places and nowhere else. And that's, I think, a profound difference from earlier technologies that relied on random changes that could not be controlled. So it's a really important difference to understand. And I think it does open the door to very important types of manipulations of plants that will lead to better agricultural products without some of the risks, honestly, and the time involved from earlier technologies.
Thank you. Thanks very much. And thank you, Mr. Paolo. Now, we have some questions coming in, which means it's unlikely for me to get my question in, but let's see where we get to. I have Kai Pernhagen. I have some others to follow. Kai, if you could start off just by telling us your affiliation and then your question, please.
Can you please repeat the name of the participant so I can upgrade him to the speaker?
Kai Pernhagen.
Okay. I can't see him. Kai.
In that case, we'll come back. Can we go to René Custers next?
Yeah, of course. Okay. Please, René, can you open the microphone?
Open the microphone. And you should also be able to see me now. Thank you.
Yes, we can.
Yeah, thank you. I'm immediately going to pick up on the point you were just raising in terms of the speed that CRISPR is adding to breeding. Most of the edits that we perform are similar to the types of changes that can occur spontaneously in nature or result from conventional breeding, and that is a very important point here. But the speed is of course the acceleration of the breeding process is very important, and what you can also do, of course, is that you can simultaneously alter multiple genes, or in a short period of time stack quite a number of different small changes that you can introduce using CRISPR. And I think some of the criticism that is being voiced by a number of people in the organization is related to exactly that point. The fact that it may go very fast in the number and types of changes that we may be additionally introducing into crops. Do you, as such, perceive that as being a risk or not?
Thanks, Professor Doudna.
Thank you for the question. I would circle back to what we said in response to the original, the first question, which is that CRISPR accelerates the rate at which changes can be made, but in that regard, it's basically accelerating the pace of what is otherwise a natural process. So to the extent that the choice of genes to be altered maps to known functions, and that's typically what is being done with traditional plant breeding, CRISPR is simply making it faster and safer and more precise to make those types of multiple changes.
Okay, thank you.
Thank you, Renee. Thank you, Professor Doudna. Did we locate Kai Pernhagen?
Yeah.
Okay, good. Please go ahead. Thanks. And maybe you could just tell us where you're from. I think that helps.
Dr. Kai, can you please open the microphone?
Yes, I can. Can you now hear me?
Yeah, we can hear you, please.
Excellent. Thank you very much. My name is Kai Pernhagen, professor for food law at the University of Bayreuth. And thanks so much for the amazing speech, Professor Doudna. I have a short question. I mean, there's currently a hearing going on from the Commission about the new regulation about CRISPR. And one of the frequent criticism that is raised from particularly people who would like to see CRISPR regulated really heavily is that, and I will quote, CRISPR can persist in cells for weeks, bouncing around the chromosomes, producing damage to DNA over and over again every time it finds a near match to the intended target. How would you respond to these criticisms that, for this very reason, argue for a strict regulation? Thank you.
Professor Doudna.
Thank you for the question. The way that the CRISPR technology is deployed is important here with regard to your question. Most of the work that I'm aware of that is ongoing at our institute and with our commercial partners, for example, uses the CRISPR protein itself. And that means that that protein has a short lifetime in cells. And once it does the initial editing, it is rapidly degraded and destroyed. And importantly, that also means there's no remnant of the CRISPR editor that hangs around in the cells over time. So this is a really important thing to understand because it does mean that there is a short timeline for the editing to take place, after which it's finished.
Thank you very much.
Thank you. Next, I have Luca De Biase.
Okay, give us a second to unlock Mr. Luca De Biase. Okay, please, Luca, now you can turn on your microphone and webcam.
And Luca, if you could just give us a little bit of background, who you are.
I'm just a journalist. I'm working with Reimagine Europa for our projects about media ecology, which means that we are worried or engaged in helping the media become less polluted, polarized, and incomprehensible. So this is what motivates my question is, of course, the fact that we are in the middle of a big debate about this, and it has a history of bad debates, because we did in the past debate about some advancement in science that we really didn't understand very much. So our choices were not rational enough. But how to make them more rational is a problem. In this case, of course, listening to Jennifer Doudna means to be much more safe and calm about what is going to happen. We have seen her work for the moratorium about the use of CRISPR for humans, Genome, and we have seen how her job has been done with a lot of responsibility on this. So my question is really such an open question that you may smile, but I would like her to ask the question that we should make to us in order to put the problem of CRISPR in agriculture in the right way, so that we can solve it for ourselves, our society, our media environment, and our political environment.
Thank you, Luca. Professor Doudna.
Thank you for that comment, and I think you're asking me to ask a question. And if I'm understanding correctly, I think what you're getting to is the fact that, again, CRISPR is a transformative tool. It truly is a new way to work with plants and with microbes that can affect agricultural products, and to do it with precision and with the kind of accuracy that I think actually leads to more safety. So I guess the question to the European Union and to all of you is, how do we as human beings want to see that technology deployed? And how do we make sure that that deployment is safe?
Thanks very much. Thanks, Luca. Thanks, Professor Doudna. I'm going to mention the next three names I have on the list to make it easier for the tech people to find them. So next I have Julia Sika, then I have Peter Chase, and then I have Heis Klater. So Julia, please. Okay.
Okay, please, Julia, can you turn on your webcam and microphone?
Hello, and thank you. I'm a science journalist working for the Austrian newspaper Der Standard, and I would like to ask the question that the way CRISPR is used now in labs could probably be referred to the first stage of a new technology. What do you think? What kinds of improvements can we expect? And another question that goes more into the ethical direction. A lot of the criticism against GMOs seems to be criticism of modern agriculture in general, how few big corporations control the food supply, etc. How could this discussion be conducted better and constructively?
Thank you, Professor Doudna.
Thank you very much for those questions. To your first point about improvements to the CRISPR technology, it's extraordinary how fast the technology is advancing. I do want to emphasize that point. And the kinds of improvements that are happening right now include increasing the number of genes that can be manipulated in a single experiment, especially in plants. This is a very, very powerful mode of using CRISPR for making multiple changes in a single experiment. We're also advancing ways of using CRISPR in more and different types of plants, not just the major crop plants, but also increasingly the types of plants that are grown on small farms in certain parts of the world, in backyard gardens, that sort of thing. The tomato is an interesting example of that, I would say. And it's also increasingly possible to make changes that allow insertion of DNA, not just disruption of a DNA sequence in a plant, but also insertion of new genes, again, with the kind of precision that only CRISPR allows. So those are all things that are happening now, and I think over the next few years we'll see extraordinary advances in the accuracy and efficiency and precision of CRISPR in plants. And your second question regarding large corporations that have traditionally dominated agriculture, at least in certain parts of the world. Honestly, and this is not my area of expertise, but one of the things I've found most fascinating about using CRISPR in agricultural settings is the fact that I think it's actually going to be a democratizing tool, and it will make it possible for many more players to be participating in the agricultural space. Small companies, small farmers. Why is that? Well, it's because of the nature of the CRISPR technology itself. It's a truly enabling tool, and a reason for that is that it's not difficult to deploy it. And so that's been one of the reasons why it's been adopted so quickly over the last decade. And also the reason that more and more people are utilizing it is that it's widely available. It's not sequestered in a few hands, in a few large companies, but in fact, it's available to everybody from new entrepreneurs, new companies, big established companies as well, but also to many people who otherwise wouldn't have access to a technology like this.
Okay, thanks very much. Thanks, Julia. So next on my list, I have Peter Chase. Peter.
Yes, please, Peter.
Okay.
Hi, can you hear me?
Yes.
Okay, I'm Peter Chase. I'm a senior fellow with the German Marshall Fund. And I'd like to, your book, A Crack in Creation, is about an ethical journey about the germline editing. And implicit in that is that you need to draw boundaries. A feeling that boundaries need to be drawn around technologies. Technologies are not moral. They're not good or bad. They can be used for both. What you just said underscores that this is a technology that can also be used, maybe very precisely, for not necessarily good outcomes. So I'd like to wind you back to the question that you phrased in response to how does a regulator determine whether or not a certain change is an acceptable change? Whether or not, and how do we set up a system so that democratized technology that can be done by high school students sitting in their lab is not abused? I mean, it's a powerful tool. And so how do you make sure that this is a powerful tool that is properly used?
Professor Doudna.
Yeah, that is a profound question that you're asking. And it's one that I have been grappling with for basically ever since the first work that I did with Professor Charpentier. And I guess the way that I think about it, at least in the applications that we're discussing here, is that it's a technology that accelerates what would happen naturally if we had enough decades or centuries to wait for traditional plant breeding to take place. And so to the extent that natural changes are happening, it's just that we're using a technology to accelerate the rate of those changes. I think that, to me, that seems like a one way to view what can be considered to be safe and allowable versus something that is, really not part of what would happen in the natural world. And as you know, both types of changes are possible with CRISPR, but I think that, the way that agricultural products, crops are being manipulated with CRISPR, I think, to me, starting with what can be done that is simply accelerating the process of nature is one way to think about it.
Okay, thanks for the question, Peter. Thanks Thanks, Professor Doudna, for the response. Next, I have Gijs Kleiter, and then we'll go to Erica. Gijs, are you with us?
Yes, I am.
Okay, good.
Okay, well, thank you very much also for the excellent talk. One question I have is actually, you mentioned the application of CRISPR-Cas in plants, and I think plants are special because there we have a history of what we call mutation breeding, which are relatively imprecise methods. But also, I'm from Wageningen University Research in the Netherlands, and our government actually has made the case for the application of CRISPR, not just in developing plants as such, but also in research. So actually the research stage where you actually try to create variability within a particular crop for which you may not have very much resource materials with genetic variability available, but in order to be able to investigate the impact of certain mutations on the performance of the crop, perhaps even without using CRISPR to produce the final crop, but at least to assist breeding. So what I'm trying to say is that actually there's a lot of talk about comparing CRISPR with transgenesis, but I think in the case of plant breeding, there might also be, a comparison with mutation breeding. And what's your take on this? So we have this old-fashioned method of mutation breeding versus the method you helped to establish. Well, thank you very much.
Thanks, Gijs. Professor Doudna.
Yes, well, this is an important point. Again, I would say that CRISPR is a tool that accelerates that process and makes it much more accurate and precise. And that comes with, I think, important advantages when we think of applying it in agriculture, again, with appropriate regulatory guidelines in place.
Thanks for the question, Gijs. Thanks for the answer, Professor Doudna. Erica.
Well, thank you very much. I have a lot of questions, but I'll just say the first one. You mentioned in your presentation that this summer we've seen a lot of extreme weather in Europe with floods on the one side, extreme heat waves and fires on the other. And this has led to crop failure, new diseases and a lot of problems. And with unpredictable weather set to increase in the next couple of years, as opposed to we're starting to see a changing debate in Europe, with President Macron starting to really say, how do we rethink words like tradition and terroir in Europe? And my question is, do you think that a technology like CRISPR can actually help preserve our traditions and our the traditional kind of agriculture in different regions, and that actually part of the kind of the fact that it can be so precise and worked on very also on small scale spaces, it can also be very local, so you can actually adapt to very local problems as opposed to having to find solutions that fit kind of a one model globally or at least regionally.
Okay, thanks. Professor Doudna.
Thank you for the question, Erica, and I could not agree more. I think that's something that I'm very excited about with CRISPR, is that it truly does provide the kind of opportunity that you just said, namely that one can imagine making changes to plants that are growing regionally, responding to local changes, environmental or otherwise. And this is something that is enabled by a tool like CRISPR because of its precision and its accuracy with editing. And furthermore, with regard to maintaining crops that have been perhaps cultivated over hundreds or even thousands of years, the thing about CRISPR is that it allows maintenance of all of the traits that we've come to appreciate in a crop, while perhaps introducing one property that might now make that plant better adapted to dealing with, say, drought or extreme heat, without changing other features of the plant that we appreciate.
Thanks, Erica, for the question. Thanks, Professor Doudna. I have just on my list one more, and I think You've done a lot of hard work with your presentation and taking all of these questions, and I think we should probably leave it at that. So I have Petra Jurash. Petra, are you with us?
A bunch of seconds. I will upgrade Petra to show him to make the question. Peter?
Thank you. Petra?
Peter Chase, right?
No, Petra Jurash.
Ah, Jurash. Okay. Okay, please.
Yeah, hello, good afternoon. Thanks for this excellent presentation, and I really enjoyed it. So I'm a representative from Euroseeds, which is the European seed sector representation. And my question is regarding the accuracy of the technology. So we hear a lot of criticism that the technology is not that precise as, let's say, promised, and that there are a lot of off-targets effects that might create potential risks also when we apply the technology in plant breeding. Maybe you can comment a bit on that also in the context of maybe earlier breeding methods and how you look at the potential risks of those off-targets. Thank you.
Thank you for the question. The accuracy of CRISPR is something that has been investigated extensively over the last 10 years. And I think the field has come to understand that although no technology is perfect, CRISPR is intrinsically quite accurate, and it's been possible through engineering of the protein itself, as well as other ways of controlling its activity. For example, careful selection of the actual target sequence in DNA that gets edited. It's been possible to improve on that natural accuracy of CRISPR. Is it important to continue those types of investigations? Absolutely. And one thing that's happened over the last 5 years really is that there are increasing standards in the field for assessing accuracy of editing, for techniques for looking at the intrinsic accurate versus off-target edits that might happen during an experiment. So even when there are off-targets, it's possible to detect them and then not utilize those cells or plants. So and I think we'll continue to see very rapid developments along those lines in the coming years, because that is an area of great interest in the field.
Thanks very much, Petra, for the question. Thanks very much, Professor Doudna. I have the feeling that we should give you a break now, because I know it's early in the morning for you, and I know that will be a major challenge for me. But thanks so much. Thanks so much, and we hope to meet you again soon. Oh, I have just one more.
Please.
Ava. Do we have Ava? I'm sorry, I am not really expert.
Yes,
at
we
these.
have Eva.
Yeah, I'm not really an expert at these Polish names. They can trip me up. Ava, please. How are we doing? Ava, are you with us?
Can you listen to me?
Yes, yes,
Yes,
we can hear you.
Thank you. My name is Ewa Lojkowska. I am from the University of Gdańsk in Poland. I have a question. Because majority of crop plants are polyploids, do you think this is a problem for this technology, for the CRISPR-Cas to obtain plants with the new features?
Thanks for the question, Ava, and thank you for keeping it short.
Wonderful. Thank you for the question. I'll try to keep my answer short. The short answer is I don't think it's a problem. And the reason is that the CRISPR technology is quite efficient. And so what's been found is that the molecules doing the editing are typically able to edit all alleles of a gene in the cell, although that is something that has to be monitored during the application of CRISPR.
Okay, well, thanks very much. And really, once again, thanks, Professor Doudna, for taking your time. And I hope we will see you again at a certain point. So thanks so much.